Okay, GM algae for biofuel production, biosafety and risk assessment. Here we are going to deal about the production of biofuel, how a genetically modified algae helps in the production of a biofuel, and what are its safety and risk assessment terms. So let's proceed. Well, at the end of this presentation, we will have a brief knowledge on what are biofuels, how is biofuel generated from algae or GM algae, genetically modified algae, what is or how is a GM algae actually generated, and what are the biosafety and risk assessment concerns relating to biology, as well as the status and possibilities in the context of Nepal for GM algae. Well, let's go and see what are biofuels. Biofuels are anything like bio-oil, bioethanol, biomethanol, fuel, biogases, consumption production, biomasses. All of these things are biofuels, as you can see in the figure here in the right side, in the right column. Yes. Okay, so this biodiesel, this biogas, which is common in our villages, and this is briquette, and as well as the woods are also biofuels. So these are all the biofuels, and today we are going to discuss the biofuel from algae, how an algae. Okay, why did I choose microalgae? Why did I choose algae for production of biofuel where we have all other biofuel resources? Why I choose microalgae? Because it has a short life cycle. As you can see, algae are minute creatures or minute plants that have a very short life cycle, that do replicate and that double in number within every 24 hours, or some strains even 2 to 3 times in 24 hours. So it has a very short life cycle. Perform photosynthesis. They are green, so they do produce the they do perform the photosynthetic reaction. They're autotrophic and they can synthesize their own food. The other is they utilize the saline and wastewater for growth. One of the most important characteristics is they use saline water, so we can use the polluted water as a nutrient source for algal growth, not arable land. Why? Because when starting with the biofuels, like we deal about different soybean oils, different biofuel products like canola and jatropha's, will then need to be cultivated inland, and they use huge and abundance of cultivable land. But in case of algal, it doesn't need any arable land because it uses water, carbon dioxide and light for preparing the food, so it doesn't require an arable land for cultivation. And last and not the least, which is useful in carrying electricity and in transportation, as we are all facing the fuel crisis nowadays. We are all known how much a fuel is essential for transportation. So algae, the fuel from algae, is totally essential for transportation purposes as well. So that is what I chose microalgae. And as you can see in the right side of this figure, there are different kind of products that can be generated from microalgae. That one of them is animal fuel, the others are fish stock, bioplastics, biofuel, cosmetics, nutraceuticals or pharmaceuticals, vitamins, foods. So there are multiple advantages of microalgae. Now, only why did I choose microalgae? One was the advantage of this, it had beneficial regarding the other products, and the other is the production or yield of oil from the bioalgae. As you can see in the chart here, from corn to microalgae, in the 8th number, the production of microalgal oil is 5,000 to 15,000 gallons of oil per acre per year, while on the same, if you see at the corn, it's just 18. And Jatropha curcus, that has recently been categorized as an essential product for oil production, for biodiesel production, it just produces 202 gallons of oil. So regarding the yields, microalgae is our best fit and a best choice as well, a smart choice. So if you can see at the overall data, or how the overall diagram of how the algae produces biofuel, you can see algae is cultivated. The carbon dioxide generated from the factories, because it is a major byproduct of multiple industries, and that is causing a massive air pollution. So these can be used as a potent nutrient source for the cultivation of algal. As you can see in this figure, the algae utilize carbon dioxide, sunlight as well as the water, and it synthesizes the different products, including the oil. The oil produced is harvested and extracted, and then further it is refined in the factories, and finally that can be used as a jet fuel, gasoline or diesel. So This is a simple overall diagram of how an algae produces the biofuel. So let's go in detail what's inside it. Okay, now if we talk about, I have just been saying about algae, and now I'm going to talk about the GM algae. I'm focused on GM algae. Why GM algae? Because the microalgae I talked about producing about the yields is all about some particular strains, and not all the strains of GM algae can produce the sufficient quantities of oil that is required to supplement to the fuel industry. So I choose the choice of GM algae is the best and the most perfect choice to enhance the productivity of the GM algae. So genetically modified algae. What is the status of GM algae? If we talk about the different types of algae, there are two basic types of algae, that is green algae and cyanobacteria. Among them, green algae is important organisms that and have a potent capacity for biofuel production, and the status, as you can see in the figure, the chlorella, denaliella, nanochloropsis, syndesmos, botricoccus, neochloris, and purachlorella. So these have all been brought a genetical modification, because this is a genetic era and genetic engineering has taken peak. So we are trying to focus on how that can be used for the prevailing fuel crisis. Chlorella and denaliella as seen here, the hydrogenase enzyme or the gelanase or galactosidase, phytase, phosphate and hydrolase and manase enzymes have been genetically modified to bring an increase in yield or productivity of the oil in green algae. Well, the most frequently used trace is chlamydomonosterin hardy, that has been used for many different traits. But blue-green algae, in comparison to blue-green algae, it does produce oil maximum, but in comparison to green algae, the blue-green algae doesn't produce much oil. Instead, it does fix the carbon dioxide more than plants, but the capacity of blue-green algae to undergo recombination is too high, and it is too much unstable, and it is series genes too fast, so that this is not a choice. But though it has been multiple used because of its capacity to interchange its genome, and it is so easy to perform genetically changes in the blue-green algae. So this has been a potent choice, although not a higher rel. Instead, we can engineer the genetically engineers gene produce higher L. Synechocystis PCC6803 and Synechococcus elongatus PCC7942 are the two major strains of blue-green algae that have been used, and as you can see, as in the various acyl carrier proteins, reductase and dialdehyde formulation in oxygenase alkanes, and acyl carrier proteins, histidine, KIVD and ADH from Lactococcus lactis. The KIVD and ADH from the Lactococcus lactis that is used for production of isobutanol has been engineered into Synechocystis for production of the bioethanol. So in this way, it is not a short fact that which gene is to be modified, but the thing is what product are you going to develop. Like it may be ethanol, it may be diesel, or it may be lipids. Whatever product you are going to derive, you need to find a potent organism, so potent gene for that production, and you are just engineering it into the blue-green algae. So it's not that simple, but it is that way in a simple way. Okay. Now, as I said before, yes, the green algae I've talked about. Green algae undergoes, as you can see, it undergoes about 24,000 changes between two common laboratory strains, and in case of blue-green algae as well, first organism to be genetically transposed, and this reported to be silencing. So it was found that green algae does have a capacity to generate higher yields, but in comparison to blue-green algae, it is too much transformable. So this is what we found. What we found was a strain found in our laboratory, the same strain in the other laboratory did show multiple differentiations in their characteristics. So these ever-changing characters, instability in the genome, was a major demerit in choosing green algae, and the same was the case with the blue-green algae as well. There was silencing of the gene, like as shown in here, N2 amino acyl glycine. If our gene that is transcribed, or that is generated, or that is modified in the host is simply silenced, and there is no motive of gene production or genetic engineering. So that was also seen as a major potent disadvantage of this genetically modified algae. And now, regarding the mode of transescape and integration is of course sexual reproduction in case of green algae. Gametic differentiation triggered in Chlamydomonas hardy by lack of nutrient or moisture. So sexual reproduction did occur in green algae, but in case of the cyanobacteria or blue green algae, there was no genetic transfer. There was no sexual reproduction, but there was only horizontal gene transfer. So in case of green algae, there was lesser chances of horizontal gene transfer and lesser chances of transgene escape. But in case of cyanobacteria that we chose as a potent carrier for our gene modification, for our genetic modification in our algae, was so a higher rate of genetic modification. As I saw, it had a potent capacity of interchanging of genomes, so there was a horizontal gene transfer. So there was a risk that the gene we incorporated could escape into the environment, and it could invade the natural flora and cause a pollution in eco pollution. So that was also a risk to us. Now, how did we contain our GM algae? Containment of the GM algae could be done. Now, there were two methodologies where how genetically modified algae could be used for the cultivation for the production of the biofuel. First was enclosed system or photobioreactors, and the second was outer open ponds. Outer open ponds, so though did produce a higher yield, though had a they were cheaper, but the problem was it got contaminated, and the water sources could be mixed up with the other water sources and could create the pollution. In case of the enclosed bioreactors, yes, there was a less chance of pollution, and everything was in the controlled environment. Everything, nutrients, sunlight, water, everything could be provided in a controlled environment, but it found to be costly. So that is what containment. That is how there was containment facilities, and we preferred bioreactors to outer open ponds. In case of open ponds too, the dams were made higher water levels for controlling the transfer of GM algae or mixing of GM algae with the outer environment. And the third thing that was done was genetic approach. In case of genetic approach, I have discussed about that in GERT, like the genetic modification was done in such a way that after the requirement of the synthesis, the gene was silenced, and as it went into the outer environment, that got silenced, like variety got and trait associated got we'll be talking about. What it does is, after the sufficient amount of product is produced, it did cut off its gene and silenced its own. So gene silencing for the outer environment is to be made to contain the genetically modified algae from mixing into the environment. And this is a simple diagram of a photobioreactor in the left, and on the right there is an open pond reactor where the algae has been cultivated. Okay, biosafety and regulatory aspects of genetically modified algae. So regarding the biosafety and regulatory policies, there are multiple regulatory policies and risks. Now, agrobacterium, as you all know, for genetic modification, agrobacterium is used, and agrobacterium itself is regarded as a pest. So how would any organization or USA itself won't give permit to market or make the thing that is generated from a pest, because there are risks that the agrobacterium genome too may get characterized, and that may involve or infect the surroundings. So that is a major risk. The second is GM regulatory body, and there are multiple GM regulatory bodies that are institutional biosafety committees, state GM regulatory bodies, national GM biosafety review committee. All this should assure that our GM product is actually of no harm to mankind. So there are multiple steps and multiple institutions. There are only three for international level. For national level, there are still multiple organizations you have to go through, and it takes a long and huge procedures. And what are the risks? How do you characterize the risk? Toxicity. Well, while characterizing the risk, the major thing is toxicity. That should not be toxic to any plant or animal. So that is the major thing. Second is antibiotic resistance. In order to make genetical modifications, we use antibiotic resistance marker so that we can characterize the genetic modification has actually taken place. So these genes may bring about an antibiotic resistance in the certain traits that can mix up with the pathogens in the environment and can further cause a massive destruction in the ecosystem, so that should also be taken care of. Therefore, there is a competitive advantage on the recipient organ. The Intergovernmental Oceanography Commission should this commission, we can know which are the resistant, which are pathogenic, which are toxins. All this we are to first know about if our strains are free of all these risk factors. Now, hazard assessment, like as you can see in the figure, small Prototheca vickers, there are multiple strains of algae that does cause an infection or that can cause blooming or signs of disease in allergic reactions to the people, so that the algae are used, the algae generators should be free of toxins like cyanotoxin, neurotoxin, hepatotoxin, cytotoxin, endotoxin, etc., so that could be of no harm to human. Exposure assessment, yes, this is the most, and I have been repeating it from the beginning of the slide, that it should not spill into the natural ecosystem and should not rupture the natural ecosystems. So there must be multiple detection, sophisticated multiple detection too, so that the GM strains can be identified. This is the major chart flow of how a genetically modified algae is characterized and come to market for a larger scale. Like first, an application for a GM algae is signed. If we have generated a GM algae, the information provided is per the GMO guidelines. Then the state level GMO regulatory agencies characterize them. If the risk assessment is done, if it is free of genetical if risks and it has no harms to human, and then national GMO Biosafety review community look after the report, and if there is no risk to the natural ecosystem to the humans, then it is confined, it is for a trial for 1 year and field trial for another 1 year, and only after that they issue the license for the commercial release of or onset of the GM algae. If not, if the reports prove that it has some risk to the natural ecosystem, it is just rejected. So in that way, this should all go in a flow after step by step in a legal and authorized way. Now we'll see the consumption in Nepal. In status in Nepal, if we see the fuel consumption is very high. The renewable energy resources have very small market, while commercial fuel and traditional fuel occupy a large scale. That is, the petroleum, the diesels, the gas have consumed the most of the entire market, more than 80% of the market, and there is a high fuel consumption. And the import of oil by Nepal Oil Corporation has been tremendously increasing year after year. That was year after year. Still, it is insufficient to supply, and now the uncertain this strikes over has all prevailed us to a very devastating and very bad situation. We are facing all the troublesome situation, and the problem we have actually realized how it feels to have a fuel crisis. We have all been looking at. And this is all the cell diagram of NOC, and if you can see in this, every year the Nepal Oil Corporation has been bearing a loss of millions of dollars, billions of dollars in oil, and it has accepted, or it has planned to make a profit of about $18 billion in the coming year. But now we have all seen what the country is facing through in this year. The strikes and the black marketing of fuels is never gonna make it. So a biofuel from algae can be our smart choice in this regard. We can use the biofuel from algae. In a way, we have been using the polluted water resources to feed the bioalgae, and the other way, the bioalgae generates the oil that can be sold to the consumers in a cheap and reliable prices. So these can be an efficient and a most active and our smart decision to make. This was a literature review. I got through some article that they had planned of generating some plans of generating algae from GM. Algae, it's not a GM, but a simple algal biofuel production, but I didn't think that it worked out well.